Good afternoon and welcome everyone. My name is Sierra Lamonaco and I am the Alumni Relations Officer with the Faculty of Applied Science and Engineering and the Program Lead for the Skule Lunch Learn. I would like to thank all of you for tuning in for today's event. With alumni tuning in from all over the world, we have guests joining us from 5 different countries, including Canada, Israel, Hong Kong, South Korea, and the United States. Now, before we start today's presentation, I wish to acknowledge the land in which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still home to many Indigenous peoples from across Turtle Island, and we are grateful to have the opportunity to work on this land. Now, a quick housekeeping item. At the end of today's presentation, we will have time for a formal Q&A session. Questions for Professor Alison McGuigan can be shared in the Q&A button at the bottom of your Zoom screen, right there. Now, for the individuals who submitted their questions during the registration period, thank you for pursuing your curiosities and sharing your insightful questions with us. Now, before we introduce our esteemed guest speaker today, I would like to take a moment to introduce and talk a little bit about longtime U of T engineering volunteer and chair of the Skule Lunch Learn program, Lori Hevola. Now, I'm going to quickly just share my screen for a second. All right. So last week, the U of T engineering community celebrated the Engineering Alumni Network Awards, which serves as a platform to recognize and celebrate our alumni for their remarkable achievements. This year, Lori was the recipient of the Malcolm McGrath Alumni Achievement Award. Lori, a graduate from the class of '65 in electrical engineering, has been an inspirational figure within her community for many, many years. And it's not just the official roles or the titles that define his contributions, but his ongoing spirit and his commitment to U of T engineering. From volunteering to setting up awards to support future generations of engineers, Lori is a true champion of U of T engineering, and we are so incredibly thankful for his support. Lori, congratulations on your well-deserved award. And with that, it is time for us to introduce our guest speaker for today's event, who also happens to be this year's deserving award recipient of the EAN Mid-Career Achievement Award, an accolade reserved for engineering graduates who have earned respect within the profession as well as the broader Canadian community. So I'm going to pass the mic to Lori now to introduce her and share more about her remarkable achievements. Lori, the virtual floor is yours.
Thank you, Sierra, first of all, for your very, very kind introduction and quite unexpected remarks about my award. It is indeed my pleasure to welcome our guest speaker today, a fellow award winner, Professor Alison McGuigan, who will discuss the revolutionary potential of artificial tissues in drug development, personalized therapies, and predictive disease detection. Dr. McGuigan is a professor of chemical engineering and applied chemistry, and also a professor at the Institute for Biomedical Engineering, both obviously here at the University of Toronto. She obtained her undergraduate degree from the University of Oxford, her PhD from the University of Toronto, and completed postdoctoral fellowships at Harvard University and the Stanford School of Medicine. She has made pioneering contributions to the engineering of tissue models to explore mechanisms of disease and regeneration. Her team has established methods to generate multi-component tissues with specified organization and design tissue platforms for smart data acquisition, with a focus on stratifying heterogeneous bulk data by cell population, spatial location, or time. And now, without further ado, please join me in welcoming our guest speaker, Professor Alison McGuigan.
Thank you, Lori, for the really, humble welcoming. I am very grateful to get the award, and I'm really excited to share some of the work that my team's been doing here at University of Toronto, sort of in the last 10 years. So what really is one of the key problems that motivates us is that the Canadian population is aging. So by 2030, 22.5% of Canadians are going to be over the age of 65. And this is really going to produce, it's already producing sort of healthcare expenditures that are becoming quite unmanageable. And so, why is healthcare so expensive? A large component of healthcare costs are due to the development of drugs, delivering drugs to people, and the cost to develop those drugs, the cost to decide which drug to give which person, and is it effective or not. Okay, so why are drugs so expensive to develop? How do we currently develop them? So the drug development process, this is a simplified version. It takes many years, so approximately, 10 to 12 years minimum, and it costs a lot of money. So this $1.7 billion is for one drug, but when you take account of the drugs that don't actually make it to market, it's more like sort of $10 billion to develop one drug when you allow for that effect of drugs that don't make it. But essentially, the process is you want to identify a compound that has some therapeutic effect. So you give it to a person and it helps make their disease either stop progressing or improve symptoms. So you identify this compound. Then you have to check that it's not got toxic side effects. So you do this in animal models or sort of models before you get into people. And then you basically do the reverse with clinical trials. So you do a phase 1 clinical trial to check that this drug is safe in people. And then when you get to the end, then the phase 2 and 3 is looking at, is that drug actually effective in the person at doing what you expect it to do and helping the disease? So if you think, we can think about the models that are used. So to identify these potential targets that the drug is going to bind to in the cell and have some effect, you can do this in various ways. Maybe you use patient material, you use mice results, you use cells, but ultimately some target with some evidence from probably all these models is selected. Then you do a lot of chemistry in very, very simple systems, often in these, this is a plastic dish here. So it's a 2D surface that you can grow cells in just in 2D on plastic. And so the benefit is that you can go to very, very high throughputs to optimize your chemistry. And then you go into these preclinical models. So some are these 2D plastic dishes and some are animal models. And then finally you get into humans. Okay. And so why is this drug development process so inefficient and imprecise? Because we need all the steps to make sure that we're developing a compound that's effective and safe. So we can't really take out any of these steps, but why is it so sort of inefficient and cost so much? And the reason is that these models that we use before we go into people really are not predictive of what's going to happen in the person. Okay. So, if we could get better information from these models, we could fail early. And, before we've gone into the really expensive part of the development process, because we would have better predictions of what was going to happen in the person. Okay. So why do these models not predict what's going on in the person? And the reason is that cells are kind of like people. They care what environment they're in and who their neighbors are. So you could imagine the way you dress and the way you behave is, I presume, different at work versus, say, in a nightclub. So in a nightclub, you're going to have different neighbors, you're going to behave in a different way, you're going to show different things, you're going to wear different clothes, like cells sort of show different proteins on their surface. So the environment really matters. When you take a cell out of one environment and put it in another, it's going to behave in a very different way. And so that is a problem with these models because tissues are not 2D plastic surfaces where the cells really are only connecting in one dimension. There's no gradients. Often there's only one cell type, whereas in a tissue, there's many cell types. There's blood vessels, there's gradients of oxygen and waste. So this is a much more complicated environment than a 2D plastic dish. So, this is why this is not predictive, right? So one could ask then, well, why did a whole industry develop on using these 2D plastic dishes? And the huge advantage of them is that they're very, very easy to analyze. You can add a chemical and get a readout of how the cells are behaving, or you can use microscopes and image everything very, very easily because it's, a single layer of cells. So about 10 years ago in my lab, we really started thinking about this problem where when you design a tissue to test a drug or think about identifying new drugs, you really want to balance the complexity of the biology that you can model and the relevance of that biology with the ease to acquire data from the system. So can we be smart about how we assemble these systems to really allow us to acquire very useful data from them in an easy way? And so if you think about our 2D monolayer, it's very easy to acquire data, but the complexity of the biology that you can model and its relevance to the person is very low. When you go to, say, a mouse or another animal model, one could argue certainly the complexity that you can model is better, is more complex. The relevance, it's a debate. It's also a mouse. It's not a human. So there can be species differences. But, people go up the scale of animal models to much more complicated animal models that are more similar to human. But, the ability to acquire and analyze and get throughput on these systems is much lower. And so these engineered tissues are really somewhere in the middle. And, the goal of my lab is, can we be smart about how we engineer these tissues to try and move us up into this quadrant where you've got high relevance and high complexity that you can model, but it's easy to use and very easy to get data out of the system. So, maybe I'll just first describe what is one of these engineered tissues and how do you make this sort of predictive human tissue, or assay is sort of the technical word. So the general principle is that you have a patient and you take some kind of sample material. So that might be a tumor cell, it might be fibroblasts from the skin that you can reprogram with certain protocols to make into a induced pluripotent stem cell. It might be tissue-specific cells, so either stem cells or specific cells of that tissue that you have protocols with certain chemicals in media that you can grow these cells. Or potentially it's things from the blood, so blood-derived immune cells. So you take your sample from the patient, you then may need to amplify that sample so that you've got more material to construct your tissue from. And so, there's protocols to potentially amplify these to make more of your tissue. And then using these engineering tools, you select which of these components you want, and then conform them into a multicellular tissue structure with very controlled composition. And I put here often in a hydrogel. So a hydrogel is sort of a jello-like material that's squishy, kind of like the material that the cell is in the human body. So then once you've got your assembled tissue, the idea is that you can read out things about either the properties of the tissue, the gene expression patterns, or actual functional behavior. So, if it was a muscle cell, how well that muscle cell contracts, or how well a stem cell repairs a piece of tissue. So this is sort of the general principle of how you build these engineered tissues with the goal of being able to sort of use them for health applications. So, there are many potential applications that are emerging for these systems. I'm going to talk about a few today, but I'll just kind of give you an overview. So the ones that really have gone furthest in terms of commercialization, in part because every drug would have to go through them, is predicting drug toxicity. So just, do you get off-target effects? So cardiac toxicity, liver toxicity, kidney toxicity. So there are certainly groups here at University of Toronto that have really focused on this toxicity. And those same systems are going to likely prove very powerful in thinking about environmental toxins. So, as we generate lots and lots of new materials, we don't know how toxic they are. We don't know how they're going to impact human health. So, are these systems that could give us some clues towards that? Then you can also use these systems to find new drug targets. So molecular components on the cell that you can interfere with a drug to produce some therapeutic effect in the person. For many diseases, they're very complex, and it's not like one target isn't good enough. So, can you find drug combinations? So that's another application that, is a really potentially high impact application, because you can scale these tissue models. So being able to sort of go through many combinations that are non-intuitive and sort of build in AI is something that's sort of emerging. I'll talk a bit about that right at the very end. Predicting patient response. So can you, use these tools to understand if there's two drugs you could give a patient, which one they're more likely to respond to? Disease progression. So, this is a bit far out there, but in the very long term, could you sort of build these models to understand, are you at risk of developing a disease? Or if you have the disease, can we predict how it's going to progress and sort of the timeline to sort of inform how that person would be managed by their doctors in the clinic? And then biomarkers, which is also sort of similar. Can you use these to get better biomarkers, where then when the person gets a biopsy or something done, there's more informed genetic information that could be taken from that material to sort of, again, tell the person and predict how their disease might develop, because the uncertainty associated with disease is very stressful. So if you can give a bit more information to people, then sometimes that can be sort of valuable from sort of a patient perspective. So in my lab, we've developed platforms for a number of tissues, mainly with a focus on cancer, some work in skeletal muscle and adipose tissue. Today, I'm going to actually just focus on the cancer models to kind of give an overview of some of the platforms that we've developed. And, why is cancer sort of a good application for thinking about these models? So there's a number of reasons why cancer as a disease is a kind of useful test case for showing the power of these engineered tissues. So one is obviously there's a huge clinical need. So, 1 in 2.3 Canadians will get cancer in their lifetime, and about 30% of deaths in Canada each year are due to cancer. So there's a huge clinical need. There's a lot of biobanks that have been established with associated patient information. So this is very powerful because you need these cells from lots and lots of different people to understand population diversity. And so in the context of cancer, these biopsies that I talked about that you take from the patient and you amplify the material, that's already been done. So it's kind of a good starting point to build these tissues. And then the other thing is that tumors are very complicated spatially and in time. They grow, they've got leaky blood vessels. So it's a there's lots of variation in the system. So it's a kind of good value problem for building these 3D models that can really try and model some of that variation. So, I'm going to talk about a number of these applications where we've made some progress towards these sort of this vision of where these human engineered tissues could really have impact in cancer. So one that I'll just highlight, which is not work from my lab, is thinking about patient response, because the material that they've used in these systems is the material that we've used to build a lot of our tissues. So it's worthwhile kind of showing the impact that's had already. So how do you make these tumor organoids? So this is sort of that material that you take the biopsy from the person, and then you have these little bits of tumor. And so the way they establish and amplify that amplification step that I was talking about is you basically take these cells, you take them straight out the patient and you mix them in this jello-like material. It's actually called matrigel, and it's extracted from mice tumors. And it's got, different growth factors and things in it, and different proteins that the cells bind to allow them to establish these organoid cultures. And then what you can do is amplify and grow up lots of these organoid cells once they're established. And then you can take these cells and separate them all so that now they're a suspension of single cells, and put set numbers into each well of this 96 well plate, so the plastic dish, and then assess drugs. And the idea is, if an organoid responds to the drug, so if you add the chemotherapy and the organoid dies, does the same thing happen in the patient? So can you predict patient response? So certainly to date, there is some evidence that this works. Okay, so for some cancers, for some drugs, there is good prediction between what goes on in that 2D plastic dish after growing them in the 3D structure. And, in some cases, the 3D model is what they add the drug to. And the patient response. So it's not perfect. So there's more to learn, and, an opportunity to build in these more complicated tumor-like structures than just sort of floating in the jello. But there's certainly some evidence that using this source of cells has the potential to sort of actually model what's going on in the patient. So, just to sort of say, these are the cells we've used. So there's sort of good evidence that they're a good source to use them for these models. So the next one I'll talk a bit about is using these cells or these models to try and predict new drug targets. So I'll talk a bit about one approach that we took. So in a tumor, I said before, there's many other cell types, okay? There's immune cells, as well as the tumor cells. There's these cancer-associated fibroblasts, so CAFs or fibroblast cells, and then endothelial cells, which are blood vessel cells. And certainly in the cancer type that we have spent a lot of time studying, which is pancreatic cancer, these cancer-associated fibroblasts are a large portion of the tumor. And initially, these fibroblasts are outside the area where the tumor is. And over time, they invade the tumor cells, secrete things that make these fibroblasts invade in. And once the fibroblasts are in the tumor, they have a very bad effect. They make the tumor much more aggressive. So they secrete things to allow the tumor cell to metastasize and move to other sites in the body. They chew up the matrix and make paths for the tumor cells to escape. And they also secrete things that just make the tumor cells divide more. So they provide an environment that really lets the disease progress faster. So, and in many cancer types, if you have lots of these fibroblasts in your tumor, so this is head and neck cancer specifically, then you have a poor prognosis. So you're likely to have much more aggressive disease. If you have very few fibroblasts in your tumor, then you have a much better prognosis. So our concept here was, can we find drugs that you could give before or with standard chemotherapy that just reduce the number of fibroblasts, stop that invasion process in, and or the proliferation amplification of the cells once they're in the tumor. With the idea that if you just can control and reduce the number of fibroblasts, maybe you'll have a better prognosis. So the system that we built to do this is called GLANCE. So the way we do this is we take a plastic chip, and I hope people can see my mouse. Maybe I need to put a pointer visible here. Hopefully you can see it now. So you take this plastic chip and you put little cavities in it using a metal stamp, sort of similar to the technology they use to make microchips. So you put this little cavity in the plastic, you bring this rubber lid down on top of the plastic, and it's lined in such a way that there's an input and an output port, and then you can inject your cells and in that jello-like material into this little cavity. So you can do all the different samples. And then when you're done, you peel off the lid and this little gel that has very specific flat dimensions, which is important, you now have an array of these little, very small volumes of jello-like material with your tumor cell in them. And then you can take this and attach it to what we call the well plate, like similar to that 96 well plate. So we use an adhesive tape that brings this down and makes it into what looks like a standard plate, which is good because then you can integrate that with all your standard instrumentation that most labs or drug screening places would have. So I'll just show you what that looks like. So this is the metal stamp that we use to create these cavities by this hot embossing. So this stamp would come down onto this plastic sheet and create these cavities. We then put the lid on, and you can see you inject here. There's these holes on either side to let air out. And we'll just see if this video plays. You can see here, it's on a cold surface so that the jello-like material doesn't cool too quickly, but you can just inject in and you get these very flat, well-defined gel regions that contain the tumor cells that you want. So, okay, so then once you've got that sheet with all the gel, you take your bottomless well plate with the tape that's been cut here, and you would peel off this white and then adhere your cells that are in these channels into the well plate, and then you've got your final chip here. And we've got this in different formats depending on the number of wells that you want. And so, you know, we can make 96 of these. You get these kind of little artifacts on either side where you injected in the gel. So you want to use computer tools to just make sure that you select the region to assess that's away from these kind of uncontrolled regions. But what's very important is that this is very thin and flat. So it makes imaging very, very easy because you don't kind of have a curved surface on a gel. So if you imagine putting a droplet of anything on a surface, you get a curve. So that's very hard to image. So just by molding between the two sheets, it's now flat. So it makes the imaging very easy. And we have strategies that we can actually make two components. So here we created a system where we put these fibroblasts on one side and then the tumor cells on the other side, with this sort of like the original way the tumor is set up, with the fibroblasts on the outside and the tumor cells segregated. So then we can watch over time, if we set it up, the tumor, this invasion process that we want to model occurs. So you can see here, this is just the tumor cells. There are those fibroblasts here, but you can't see them because we're only showing the red signal from the tumor cells. So you can see them invading. In this one here, the fibroblast is in green. You can see it's migrated to a tumor area and it's sort of moving around the tumor cell and causing that tumor cell to change and start to become invasive. So we can catch very high quality movies of this invasive interaction process because of that flat surface. So with the tool, what we wanted to do was then try and understand, can we find drugs? So we actually used kind of tool compounds, not actual drugs, but can we find compounds that reduce the number of these fibroblasts that move into this red region, or just the abundance in general? So that could occur by these cells either less moving in, or the ones that move in proliferating less, so that you end up with less total in your tumor region. So the way we did this was we took the fibroblast cells, we treated them with many different treatments for 5 days so that they were changed. We then seeded them into the gland system with, you know, the green fibroblasts on one side and the red tumor cells on the other, and then allowed 5 days for that invasion process to happen, and asked, are there ones where there was less of these green cells in the red region? And so, you know, we did this with a number of these compounds. You know, this was our no treatment in blue. So as you can see, we found kind of a number of compounds where there was a significant reduction in this invasion. So this is sort of something that we're following up on now. So, you know, that was at least a starting point of how could you find new human targets, looking at a system where it might be very hard to find that target in that specific cell population in a mouse, because in a mouse, you've got every cell type there, and often all the cell types other than the tumor cell are not human, they're mouse. So this was really sort of an example case of making a fully human system and targeting a component of that tumor environment that's not the tumor cells, that would be very, very hard to decipher if you were doing this in an animal, because there's so many cell types present. So the next sort of short story that I'll tell you is thinking about how you might predict biomarkers. So what is a biomarker? So it's really maybe a pattern of genes or some kind of feature of the tumor that predicts how the disease will progress. So, you know, if a patient went for a biopsy, they might do some analysis of the expression of certain genes to try and say, well, you have this subtype or this subtype. And so in one example here is in pancreatic cancer, if you have very low levels of oxygen in the tumor, so that's high hypoxia, is much more aggressive and survival is much less than if you have much higher levels of oxygen or low hypoxia in the tumor. So as you kind of think about this, you know, some of these markers are taken from tissue types, but we remember, or I hope you remember, the theme here that cells are kind of like people. So environment matters. So that tumor microenvironment is going to be really important in making cells behave in the way that you want, that you're going to get predictive biomarkers to actually predict human disease. So the system that we built 5 years ago or 8 years ago now, to really kind of recreate this hypoxic environment in many tumors was called Tracer. So I'll show you how this works. So this is a tea bag paper, which in hindsight actually works very well because we buy from a tea bag company and it just doesn't fall apart in liquid. So from a manufacturing perspective, it's outstanding. So what you do is you infuse into this scaffold your hydrogen, your jello-like material in your tumor cells, and you generate this sort of tumor biocomposite. So this is a strip of this tumor that you then use to assemble your bigger tumor. So to assemble, you take this strip and you roll it up around this metal coil. And what this does is it generates a layered structure. So you've taken the strip, you've rolled it up kind of like a film roll. And this region here doesn't allow any media or oxygen or nutrients to access the tissue. So the only place that oxygen and nutrients can get into the tumor is from this surface. So what happens is as the oxygen moves in, the cells consume it. And so less oxygen moves to the next layer, and the same with nutrients. So what you do is you generate this gradient similar to a tumor. If this was the blood vessel and this is deeper and deeper into the tumor, this is the culture media, so the nutrients that you give these cells when you're growing them. And as you move further and further into this stack, there's less and less oxygen available. And then you can treat the cells, you can leave this for as long as you want. And what's kind of very powerful about this system is that when you want to do your analysis, you just unroll the strip, and then from where you are along the length, you can trace back which layer you were in and which environment you were in the 3D structure. So the fact that it's a strip that you're rolling, the length along the strip really lets you register between 2D and 3D. And so you can do the analysis and sort of have the benefit of almost 2D analysis that's much easier. But you can do your cultures in 3D. So this is really, you know, this was really our first introduction of this idea, can you really design for data acquisition? Because, you know, you're culturing in 3D, but then you can do your analysis in a much simpler kind of 2D format. So I'll show you what this looks like. So this is the teabag paper. We then infuse in the tumor cells and collagen, which is just sort of a jello-like material. And you can see here the red paper fibers and the green tumor cells. We then roll this up onto this little metal coil, and we put a little plastic thing there just to hold it all tight. We've got we've kind of advanced this a lot from our first paper, so we can print in different jello type materials into the system. We've got a bioprinter, so we can print in different cell types in different patterns if we want to. And then when we want to unroll, we simply just pull the end of this, and then you can unroll the system, you can cut and collect cells from very specific locations. And because it's paper, we can actually print everything on the layers so that it's very easy to understand which layer you're coming from when you're dealing with the samples. And so, you know, we spent a lot of time characterizing this. I'm just going to show you one, I think, important result, which is when we look at cells from the outer layer, so layer 1, where there's normal oxygen, versus that inner layer of the roll, which is layer 6, which is where you have low oxygen, so lots of hypoxia. In patients, the hypoxic cells survive better against drugs and radiation. So that's really also what we see in our system. So in low oxygen, you get much higher survival. And when you treat with a drug, you get much higher viability, so higher survival in the low oxygen environment. So it's sort of mimicking these patterns that you see in different environments in the tumor, we can mimic in our system. So that was very important to show, you know, there's actually some benefit to creating this complex tissue structure. And so, you know, where this is at now, this is sort of the not quite published yet, so you get a sneak preview, is that we, you know, have integrated these pancreatic organoid cells. So from the earlier part of the talk, so we spent a lot of time just optimizing and getting the quality control on that. So we can put the pancreatic organoid cells in. In pancreatic cancer, there's a large number of these macrophages. So it's a type of immune cell. So we also now have protocols that we can collect blood, we can isolate those macrophages, and we can integrate them into the system. So you can see here the tumor cells in green, the macrophages in red. And we've done a lot, again, of characterization to show that this behaves as we would expect in patient tumors. And the result that we're sort of most excited about is that, you know, when we do our analysis and we pull out gene signatures from the material in our tracer system with the macrophages, so only in the co-culture, we've been able to define sort of a set of genes that can really separate survival in patients in patient data. So we define the signature of genes that we're going to use to analyze the patient data in our tissue model. And then when we look at the patient data, we're able to predict who's going to do better in terms of the patients. And this is really just the comparison of the hypoxia, which is a standard measure that's used now. So we're really doing, you know, as well, at least as that. So it could be a way of another tool for a clinician to sort of inform a patient. You know, it would need validated further, obviously, but, you know, it's a starting point for showing how you could use these systems to kind of augment the current clinical practices that are helping patients sort of decide how to manage the situation. So I think I've talked so far about kind of work that's been done to predict patient response. The GLANCE platform that we've used to think about new drug targets, tracer platform, which is to think about predicting these biomarkers. And for the last sort of 5 minutes or so, I will talk about sort of where we're going moving forward to think about really scaling these systems to predict drug combinations, because that's a problem that would be very hard to do in mice because you would need so many mice. So the platform that we built is almost the combination of tracer and glance to do this. It's called Spot. So what we do is we take a single paper layer this time. So no rolling up or anything, just a single paper layer. And we learned a lot about how to print polymer into this paper structure. So essentially, you're printing and you're balancing the rate that stuff comes out the printer with the rate it absorbs into your paper towel or your tea bag. And if you balance that, then you can write patterns in your paper with the polymer. So we're able to here infiltrate most of the tea bag with plastic in the format of one of these 96 well plates. And then when we do that, we can attach with our tape such that in the bottom of every well, we have one of these tea bag. We have a region that has the tea bag in it. And so the reason that we wanted to do this when we were thinking about scaling is I showed you sort of the assembly of that glance platform. And a real challenge is that you have to assemble it. You inject in the cells, but then there's an assembly step after. So that would be very hard to integrate with robots because the assembly step sort of needs alignment and you need to, check things with your eye, that it really wasn't a platform that could be used for scaling. But the principle of glance was you want a flat gel because that's very powerful for being able to image these things over time, because if it's flat, the imaging is just easier. And so the way you get the flat gel was by molding between two sheets. And then you remove the top sheet, but that removal step is what's not easily automatable. So what we realized here was that if you inject a very precise volume of this gel into this region, the scaffold kind of makes it take on this flat form because the jello infuses into that scaffold and takes on the form of the paper. In the same way, if you put a drop of liquid on paper towel and hold it up, the liquid is now suspended in a geometry in that paper towel where it's flat. You wouldn't see the drop sort of coming out of the paper towel if the volume is correctly balanced. So that's sort of the idea here, is that we can inject in this precise volume of the jello material with the cells into this preformed plate and get this flat surface. And we do have a scaffold there, so that's a trade-off with the scaling. But for many problems, that's okay. So then we can image through this single sheet and do the same kind of analysis that we could do in glance, but suddenly at scale with automatic microscopes, etc. So we're able to do this in a 96 well format. We can also do a 384, which is very, very small volumes. And this is sort of 384 and above is when you could actually start to feasibly screen for, looking at large scale libraries or medium even scale libraries. So we really had to get to this high throughput to scale the power of the platform. And so we've also integrated this with robotic, what's called a liquid handler. So you can see it here. So the instrument is coming over and getting that very small volume of jello-like material that's a liquid. So this jello-like material is like jello. When it's under certain conditions, it's a liquid. You can mix the cells in it. And then when you inject it in and then put it under different conditions, so in this case, you heat it up, it gels into a solid. So that's how you're able to kind of do the liquid part, but turning it into a solid. So we're able to automate manufacturing of the tissue and then imaging of the tissue, and we can kind of monitor growth of these organoids and add drugs and look at changes in growth over many, many days. So sort of take home message, I've talked about a number of systems. I think that these tissue models really have the potential to accelerate drug discovery. Right now, most the systems that are furthest along in terms of commercialization are to predict drug toxicity or environmental toxicity, because once you've validated your heart system or your kidney system or your liver system, every drug could potentially go through the system. So, there's a huge market for that. Where it's more looking for drugs that actually have an effect on the disease, then the model is going to be a lot more complex because it's going to be very specific to each disease. So there's a big investment up front with the hope that it's going to be more predictive of that clinical data, that you're going to get much less failure and sort of move your compounds more efficiently through this drug discovery process. I've talked a bit about, how you could use these systems to potentially predict patient response to help a doctor decide which drug to give which person. I talked a bit about the two systems that we developed that we're using to try and think about how might you discover a new drug target or develop a biomarker that would help you make predictions about how a patient might, or how to segregate patients based on how their cells look. And then lastly, sort of the efforts that we're taking right now to really scale our platforms so that we can predict drug combinations. And that's where I think, artificial intelligence, where you put the readout into some model that then feeds back in, that you can explore kind of a really large space of combinations. And, there's collaborations with the Big Self-Driving Lab initiative at University of Toronto to think about this, that you could really automate and accelerate that process to explore very non-intuitive combinations. And the goal really of all of this is to try and develop and deliver the right drugs, the good drugs that are effective to people that will gain benefit from them, to sort of make the healthcare system much more efficient, for Canadians, but globally. So I'd like to acknowledge, as all of this work is very complicated, and to do it very carefully to really hope that you're going to translate it into impact. There's lots and lots of people involved, so there's lots of funding sources. And then, this is just some of the most recent team members, but really, it's the trainees that make the magic happen in the lab. So, I stand up here and get to present all their work, but it's really, the trainees that make this all happen.
Before we close, I would like to thank Professor Alison McGuigan for being with us today. Thank you for such a thought-provoking and educational presentation on artificial tissues in the context of personalized medicine and how it serves as a powerful tool for advancing healthcare and drug development. To everyone who joined us here today, we invite you to share your feedback on the Skule Lunch and Learn series. A link to the online survey is available in the chat function at the bottom of your screen, and also will be shared with all of you after this event by email. Also, a link to the presentation will also be shared with all registered guests. We realize that many are unable to join us in person because of time zone differences, for example, so they appreciate the opportunity to view the presentation at a later date. Speaking of which, Skule Lunch and Learn events are traditionally hosted on the second Wednesday of each month. However, next month, our event will be held on Wednesday, December 6th, and features Professor Jonathan Rose, who will discuss the use of scaling talk therapy with new language, artificial intelligence. There we go again, AI. In closing, I would like to thank all of you for joining us for today's presentation. With this, I hereby adjourn today's virtual event. May all of you have a wonderful rest of your week and see you again next month. Thank you and have a good day.